Hi, my name is Angelina Crittenden. I'm a TA for the USC Young Scientists Program. I typically teach 4th grade at the Alexander Science Center School. Today, we're going to be learning about DNA structure. So, what is DNA? DNA is short for deoxyribonucleic acid. It is one of the most essential macromolecules, or large molecules, found in all living things on Earth, serving as the blueprint for the genetic organization of all organisms. Humans didn't discover it until the 1950s. DNA has a unique structure called the double helix. It looks a little bit like a twisted ladder. 2 phosphate backbones are joined by nucleotide bases. There are 4 different types of nucleotide bases: adenine, thymine, cytosine, and guanine. Think of them as just letters, A, T, G, and C, or letters of the DNA alphabet. 2 of these nucleotide bases are joined and supported by the phosphate backbone. There's an important rule though. A only pairs with T, G only pairs with C, and vice versa. I like to call AT and GC base buddies. If you have A, T, G, C on one strand, then the other strand will have T, A, C, G to match with it. These strands are complementary. Now, why is DNA important? Here are just a few of the reasons why we should care about DNA. 1, it provides the instruction manual for the arrangement of an organism and all its cells. Without DNA, the cells in your body would not survive or even exist. Next, DNA can be copied, allowing our cells to divide and create new cells. Without it, if you fell and, say, cut your knee, it would never be able to heal properly. Finally, our DNA makes us unique. We share a vast majority of our DNA with other organisms, but the small portion that is distinctly human can vary so much from person to person. You are completely unique. Unless you have an identical twin, no one on Earth has ever nor will ever have the same DNA as you. Today, we're going to make our own edible DNA models. You'll need 4 different colored gummy bears to represent the 4 different nucleotide bases, 2 strands of licorice to represent the phosphate backbone, toothpicks to form bonds between our bases, and a paper towel or plate to keep your workspace clean. Depending on what colors you have, assign different colors to different letters. For example, I'm using a different kind of candy because it has a lot of different colors. These reds will be A, these greens will be T's, these purples will be C's, and these yellows will be G's. If you don't have the right colors or enough different colors, that's okay. You can still follow along. Next, you want to write your own DNA sequence of nucleotide bases. For example, I'm going to write A, C, G, C, C, T. Now, we need to write the complementary sequence of nucleotide bases for this. If we remember the base buddies rule, we should have T, G, C, G, G, A. Now, we're going to make our model. The licorice is your backbone, so attach the gummy bears that represent the first strand to the licorice using toothpicks. So, these are my A's, so I'm going to start with this A. I'm going to take my toothpick, attach it to this A, push it all the way through, and then attach the toothpick end to the licorice strand, like that. Make sure you're careful not to poke yourself. Now, we're going to make our next nucleotide base, which is a C. We chose purple to represent our C's. So once again, we're going to puncture the C and attach that to our licorice. Next, we have a G. We chose yellow to represent G. We're going to puncture that and place it. Once again, we have a C, so we're going to puncture that and place it. Our next step is to add our complementaries. Now that I've added all of my complementary nucleotide bases, I'm going to attach the second backbone by carefully piercing each toothpick through the second piece of licorice. And there you have it, a complementary DNA strand. Now, if you're careful, you can gently twist it and you can see the double helix structure. Like we said before, it looks a little bit like a twisted ladder. And there you have it, your own edible DNA model. Congratulations, you've successfully made a DNA model. Take some time to reflect on the activity by asking yourself, what do you think might happen if there are errors made in matching each letter of the DNA alphabet? And how many individual DNA double helices do you think are in each of your body's cells? Next, let's give a shout out to our scientists of the week. Here we have Dr. Herman Branson. He was an American physicist and chemist, best known for his research on the alpha helix protein structure. His research was pivotal for understanding the biochemical roles of proteins and understanding the structure of DNA. He helped found the National Association for Equal Opportunity in Higher Education and served as president of 2 different universities. Thanks, Dr. Branson. And thank you all for tuning in. Make sure to check out our other videos.